So this is chapter 24, part 2. We ended off here on peptide structure determination. Disulfide cleavage. You can cleave disulfide bonds by subjecting it to peroxy acids. This will oxidize the sulfides, so it's no longer sulfides, and to cysteic acid, so that you can cleave all your disulfide bonds within proteins. So in terms of amino acid composition, you can use what is called an amino acid analysis or analyzer, in which you take your peptide chains, you react them with 6 molar HCl for 24 hours. That will cleave all the amide bonds so that they're all the amino acids, and then you can separate the amino acids in an amino acid analyzer, which will then monitor the intensity of the absorption as a function of time. So if we look at human bradykinin, and here is the composition of it, and then you would draw, or you would do a standard of all the amino acids, and then you can figure out, yes, this is a serine, these are proline, glycine, phenylalanine, and arginines. The other way you can determine the sequence, actually, in the previous case you're just determining the composition. To actually determine the sequence of the amino acids in the actual protein, Edmond degradation can be used. In Edmond degradation, the reaction with phenyl isocyanate followed by hydrolysis removes the N-terminus amino acid. The phenylthiohydantoin derivative is then identified by chromatography, and you can use this for peptides with 30 amino acids or less. So in terms of Edmond degradation, here's your reaction. You have your protein, your amine terminus will react with the carbon, electrophilic carbon, electrons go with the sulfur and then comes back, and then the electrons go pick up a hydrogen, generating a phenylthiourea. The phenylthiourea will undergo rearrangement, in which you have electrons from your nitrogen come in, form a double bond, and the electrons from here form a bond with a carbon, and then electrons go to the oxygen. So in this case, the phenylthiourea is protonated. So the step that isn't shown is that it's being protonated by, at least the carbonyl is being protonated. And so once you generate that, you have this cycle, which then undergoes some rearrangements, in which you have water react with the hydrogen, electrons go to the oxygen, and then this kicks this off. This forms a thiazolinone, and then essentially removes it from the rest of the peptide. So this is one amino acid, the N-terminal amino acid, being removed. In the last step of Edmond degradation, you react this thiazolinone with hydrochloric acid, and then this forms the phenylthiohydantoin, which is stable. The other method by which you can determine the sequence is the Sanger method. Sanger method for N-terminus determination. This is less commonly used than the Edmond degradation, which we just talked about, but in the Sanger method you use 2,4-dinitrofluorobenzene, which is a Sanger reagent, reacted with the N-terminus of the protein. This will just do nucleophilic aromatic substitution, and then in the presence of 6 molar HCl and heat, you will cleave off the amide bond. So this is being cleaved so that you generate this 2,4-dinitrophenyl derivative, and then the rest of your amino acids. So the other way to actually determine the sequence of a protein is to sequence from the C-terminus. The enzyme carboxypeptidase cleaves the C-terminal peptide bond, but since different amino acids react at different rates, it's difficult to determine more than the original C-terminal amino acid. In C-terminal residue analysis, you have, here's your peptide, here's your C-terminus. You're using carboxypeptidase, which will then cleave the C-terminal peptide bond, so make it into the free amino acid. And then the rest of the amino acids in that peptide sequence will also get hydrolyzed as well. Partial hydrolysis, you can break peptide fragments into smaller fragments. Trypsin cleaves at the carboxyl group of lysine and arginine. Chymotrypsin, which is another enzyme, cleaves the carboxyl group of phenylalanine, tyrosine, and tryptophan. And then you can sequence each of those fragments and then piece them together in a puzzle. Solution phase peptide synthesis is a means to actually generate proteins, at least proteins less than 30 amino acids. First, what you do is you protect the amino group in the N-terminus with a benzyl chloroformate. Then you activate the carboxyl group with ethyl chloroformate to form this anhydride of a carbonic acid. Then you couple the next amino acid. You repeat this activation and coupling until all amino acids have been added, and then you remove the protection. So in order to see this chemically, here's the benzyl chloroformate. Here's the amino acid. You're going to protect the amine group using benzyl chloroformate, right? This would be just essentially a nucleophilic attack. Electrons come up and then kick off in the presence of triethylamine. And so this will serve as the Z group, serves as a protecting group of the N-terminus. The second step is then taking the protected alanine and then activating the C-terminus by reacting it with ethyl chloroformate. So in terms of ethyl chloroformate, you're going to essentially have your electrons from your oxygen react, electrons come up, electrons come down, and then kick this chlorine group to generate this anhydride of carbonic acid, shown here. And this now activates the C-terminus, or the carboxy terminus for conjugation. Then you take this protected activated alanine, the C-terminal activated alanine, and you react it with, for example, a valine where the N-terminus is free. This will then react, right? So this is a good leaving group, so this will react with your activated carboxy terminus, and then this will kick this off, right, as CO2, and then this will come off as ethanol. So this forms an amide bond, shown here. Here's your new bond that's formed. And then you can do the same thing. You can then continue to activate the C-terminus and then couple with a free amino acid. And so this will continue on for some time, and then if you want to remove this protecting group, the way you can remove this is through hydrogenation. This will remove it, making this into an amine, and now you've just generated alanine, valine, phenylalanine trimer, or tripeptide. CO2 is released, and pH CH3. So the thing to note is that classical solution phase peptide synthesis goes always from N to C. You protect the N-terminus with that Z group first and then deprotect last. Couple each amino acid by activating that C-terminus with that ethyl chloroformate and then adding new amino acid. Solid phase peptide synthesis. So solid phase uses a polystyrene bead to attach your amino acid and then grow the polypeptide chain from that bead. Intermediates don't have to be purified. Excess reagents are washed away with solvent rinse, and process can be automated. So you can generate much larger peptides using this strategy. So you want to attach the peptide to the solid support. So here's styrene and parachloromethylstyrene to generate their polymer, which is a polystyrene, and it's abbreviated as such. And then using that polystyrene polymer, you can use a protected amino acid that has an amine protected group and the carboxy free. That C-terminal amino acid will then add on to the styrene portion of the polystyrene and then leave, the chlorine will leave. So then you've added the C-terminal amino acid to the polymer. Once you've added the C-terminal amino acid to the polymer, you can then grow the polypeptide chain. So here's protection groups for the amine terminus used for solid phase peptide synthesis. Here's ditert-butyl dicarbonate, also known as Bach protecting group. This will protect with the amine terminus. So here's the whole thing, and this portion of it ends up protecting. So what's happening here is you're going to have electrons from your amine nitrogen go, electrons from the oxygen come up, come down. This will form CO. This will cleave this, right, to generate CO2 and tert-butanol and your Bach protected amino acid. You can remove the Bach protecting group by treating it with trifluoroacetic acid. In trifluoroacetic acid, you're going to just essentially protonate. This will protonate your carbonyl over here. Once that's protonated, you then have electrons move, right, to generate the tert-butyl, the acid, right? So this is a carbamic acid. And that carbamic acid, this is going to rearrange to form CO2, right? So this will leave as CO2 gas, right? So electrons will pick up the hydrogen. This forms CO2, and then this will cleave off, and so you have your free amino acid and your isobutylene. DCC, or N-dicyclohexyl carbodiimide coupling. When you have a mixture of an amine and an acid that's treated with this DCC, the amine and the acid couple to form an amide. So you can have an acid, amine, in the presence of DCC. This will form the amide, and this will form dicyclohexylurea. In this, the first step is your acid, your carboxylate, will react with the electrophilic carbon sector, pushing the electrons to the nitrogen. Once it's activated, then your nitrogen then deprotonates from the amine. It's generating this activated and amine. Then the amine will attack, right? Because this will be a good leaving group, electrons will come off, running the tetrahedral intermediate, then electrons come back down, kicking the electrons off to the oxygen. Now the oxygen electrons will go down, and this will protonate, and this will neutralize, generating your amide peptide. You can cleave the finished peptide, so here's your peptide on your polymer, and you can cleave it off the polymer resin by essentially treating it with hydrofluoric acid and anhydrous. Because this is an ester bond, it is more easily cleaved than amide bonds of the peptide, so the amide bonds stay intact, and then this will essentially cleave. Important thing to note is that solid phase peptide synthesis also goes from C to N. You attach the Bach protected C terminus to the bead first. Couple each amino acid by removing through addition of TFA the Bach group from the N terminus. Then add the next Bach protected amino acid with DCC. Cleave with HF the finished peptide from the bead. There are different classifications of proteins. Simple is hydrolyzes to the amino acids only. Conjugated, it's bonded to a non-protein group such as sugar, nucleic acids, or lipids. Fibrous, they're long stringy filaments insoluble in water, function as structure, and then globular, folded into spherical shapes, function as enzymes, hormones, or proteins. There are different levels of protein structure: primary, secondary, tertiary, and quaternary. Primary, the sequence of amino acids represents the sequence of amino acids in a chain and the disulfide links. Secondary, the structure is formed by hydrogen bonding and forms kind of these elements of alpha helices or beta sheets. And then tertiary is a complete three-dimensional conformation. And then quaternary is an association of two or more peptide chains that have these three-dimensional conformations. So if we look at secondary structure, here is an example of an alpha helix. Here, each C double bond O oxygen hydrogen bonds to the NH, in the next turn of the helix, then forming this helical coil. In terms of beta sheet or pleated sheet arrangements, you have your sheets of polypeptides having hydrogen bonding between the carbonyl and the NH on the adjacent peptide chain, whereas in the alpha helix, it's within the same peptide chain. Tertiary structure, which is shown here, this is showing kind of this conformation with segments that have alpha helices and random coils, and it helps fold into this three-dimensional structure. And then quaternary structure, in which one of these tertiary structures can also interact with other tertiary structures to form this quaternary structure. And we've talked about the primary sequence in terms of amino acid sequences. Denaturation. So denaturation is defined as the disruption of normal structure of a protein so that it loses its biological activity, usually caused by heat or changes in pH, and it's usually irreversible. So you can cook an egg and that's denaturing an egg, but you can't go back and make it uncooked. Prion diseases is something important. Neurologist Stanley Prusner studied scrapie infected sheep brains and suggested it was caused by a protein infectious agent that he called prion protein. Prion diseases are caused by essentially misfolded protein. Misfolded protein often folded into this beta sheets assemblies. And these are essentially denatured versions of the normal protein. And once you have these kind of beta sheets aggregates, they also induce normal proteins to misfold in the same. There is Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease is a form of this kind of prion-like disease in which you have these these abnormal assemblies, and what happens is it causes this disease, and you can compare, for example, normal cells or those infected with its Creutzfeldt-Jakob disease shown here. And so this ends chapter 24, part 2.